everyone. I'm very happy to be here. First of all, I want to thank Phoebe and Rachel for inviting me. As Phoebe already mentioned, I'm Linda. I am a postdoc in the Department of Genetics in Trinity College Dublin, and I've studied during my career, my research career, human population genetics. So what I will be talking about today is the topics of Denisovans. And so what we're going to try to do is try to understand who Denisovans are. So my background is I'm a geneticist. So I know that most of the audience probably comes from archaeology. So if people have questions and want to ask questions, feel free to do if something isn't enough clear. So, okay. So first of all, let me just... Okay, I want to give a brief outline of what I will talk about today. So, as I said, we will try to answer some questions. First, we'll try to understand who were the Denisovans. So, I will give a bit of introduction, of background, of why we know about Denisovan and so what we know about them. We want to try to understand how Denisovans are related to Neanderthals and other archaic humans and modern humans. Then, okay, so we're going to go a little bit more into depth about what we do know about Denisovans and actually what we know from genetics about Denisovans. So we want to try to understand if they interbreed with modern humans, how many times they interbreed, what can we say about this, and if actually there were only one Denisovan population that contributed their genetics to modern humans, or if there were multiple Denisovan populations. Then another important part that we will try to address is the biological impact of the Denisovan introgression on modern humans. And finally, we will try to have a look at what are the current challenges and open questions on this topic. So I know I mentioned many things now, and I promise you I'm going to try to explain everything during the seminar. So first of all, so I've been invited because in the past years I've worked on this review article with my PI, Emilia Huerta Sanchez, who I want to thank for the opportunity to work on this topic. And with this article, we tried to revise, to summarize what is now known about the history of the genetic history of Denisovans, and we focused on these multiple introgression events that happened with modern humans. Moreover, I want to say that also last year, another great review paper was published by Perrin and colleagues, and in which they also revise basically what has been done in the past decade about genetic research on Denisova. So what I will talk about is mostly summarizing these two review papers, and of course I will talk about also other papers that have been published about this topic. Okay, so let's start with an introduction. So who were the Denisovans? And the first thing that we need to understand is, first of all, why they're called Denisovans. And so the reason is that there is this cave that is called Denisova Cave, found in the Altai Mountains in modern-day Russia, in Siberia. And many of you might already know that this cave is very important. It has been a rich source of ancient and archaic remains. So in the past decades, so lots of fossils have been found in this cave, and it's a very course, complex cave. There are different chambers, and as you can see here, reproduced in these figures, in different chambers there are, of course, different layers in which there have been found many remains, many ancient archaic remains. And of course, these remains are from, depending on which layers they've been found, they belong to different time periods. And the very interesting thing about this cave is that the remains that have been found are from different hominins. So in all these years, have been found remains from modern humans, but also from archaic humans. At first, from Neanderthals, so there are remains from Neanderthals, but also from these new hominin, archaic hominin that are the so-called Denisovans. So how this has been possible, why the Denisovan were discovered first, and this has been possible because of this little remain that has been found that is being called Denisovan 3. And you can see it here. So basically, this is a phalanx, a small phalanx, a finger bone that archaeologists found. And when they analyzed the DNA from these little remains, they found that it didn't belong to either modern humans or Neanderthals. But the important thing to say is that actually we don't have many remains for Denisovan. For Neanderthals, we have many remains in many parts of the world, and we will see this a little bit later. But from Denisova, we have the remains are scarce. So we have this little finger bone, we have some teeth, a mandible, and a few skull fragments. So the Denisovans remains a very mysterious population from different point of view, also from a morphological point of view, for example. So now let's try to understand how Denisovans are related to Neanderthal and modern humans. So as I already said, so basically at a certain point in 2010, scientists were able to extract the DNA from this little finger bone, and they were able to sequence it. And when they sequenced it, they found something very interesting and very important. So that means that when they built a tree, and they were built a tree with modern humans, individuals, and with Neanderthal individuals, Denisovans were not part of either of the groups. So if you see this in the neighbor joining tree, you have like modern humans here, so different population from present day different population, and Neanderthals individuals that have been sequenced. And Denisova, this is the DNA extracted from this finger bone, is not part of any of these groups. So it separates as a common ancestor with Neanderthal that is more closely related than the common ancestor with modern humans. So this was very interesting, and this opened basically a new topic, a new field, new research field, because they wanted also, of course, scientists wanted to understand, okay, but so who are these Denisovans? And actually, did they interbreed with modern humans as well as Neanderthals or not? And so on. So there were many questions that opened at the time. And what I need to say now is that this happened in 2010, and actually, so far, we only have one Denisovan genome. So this is the only one that is still available. We know that probably soon there will be another one, but still scientists now, researchers that are basically they study this topic, they need to analyze, need to do their research with only one Denisovan genome. And you may understand that this can be complicated. Okay, so now to give a little bit of context of actually what we have, not only Denisovans, but also for Neanderthal. So this is a map where you can see the dating and the geographical location of some archaic human specimens, the most important we can say. And so here you have Denisova cave. Denisova cave, we see Denisova 3, that is the one that I was talking about before. So is the Altai Denisovan, the genome that we got from the phalanx, and is dated around 80,000 years ago. We have another complete genome from Neanderthal that is called Altai Neanderthal, and it's from Denisova 5, and this is much older. So it was from an older layer in the Denisova cave. So it's around 120,000 years ago. And then very interesting, scientists found out that actually Denisova and Neanderthal interbred, and they were very lucky to find remains that when they sequence their DNA, it was the result. It's basically a hybrid, a Denisovan and Neanderthal hybrid. This is the daughter of a Neanderthal mother and a Denisovan father. So this is what we have in Siberia, in the Altai Mountains, in Denisova cave. Then again in Siberia, pretty close, we can say, to Denisova cave, there is another cave in which has been found Denisovan remains, Neanderthal remains, not Denisovan, Neanderthal, that is Chargiskaya, and they were able to, from Chargiskaya 8, they were able to sequence a complete genome, and this genome, this individual is from 80,000 years ago. And then another important cave from Neanderthal, from archaic remains, is the Vindia cave that is found in modern-day Croatia. And this genome is much younger, is from 50,000 years ago. So we call usually this Vindia and Chargiskaya Neanderthal that these are called late Neanderthals because they're younger. And these Altai Neanderthals is usually called early Neanderthals, is more far back in time. Okay, so and so you can see that this purple area here is like the geographical location of Neanderthal. In this case of Neanderthal, and here we have also Denisovans. So this is just to understand how these genomes are related to each other. So you see these are the Denisovans found in the Denisova cave. Here you have the Altai Neanderthal that is older. So it's split. The split is older compared to the one of Chargiskaya that is said it's like 80,000, and the one of Vindia that is here in Croatia. So just to give a brief overview. So another thing that actually I need to mention is that, okay, we talk about the Denisova cave because it's we can say the most important one, because we have the only genomes from Denisovan that we found is from this cave in Siberia. But actually, more recently, Denisovan remains were found also in another cave. This time, this is called the Baishia cave in Tibet, in the Tibetan Plateau, in modern-day China. And basically, archaeologists here found a jawbone, a mandible, and they were able first to analyze the ancient proteome of this from this mandible. And when they did that, they reconstructed a phylogenetic tree, and they found that these actually belong to the same clade as the one from the Denisovan in Denisova cave. So it's not a Neanderthal, it's not a modern human, but it's another branch of the Denisova clave. So this is very interesting. And also, they were able to, in further studies, they were able to analyze the mitochondrial DNA from sediments. And again, this was from Denisovan. So here we have proof that actually Denisovan didn't only was not only found in, did not only lived in Siberia, but also in East Asia. And here on the map, they seem close, but actually the distance from these two caves is like more than 3,000 kilometers. So we can say that actually the Denisovan have a broader range of where they were living. Okay, but now let's go try to understand. Okay, so what do we know about Denisovans? Did they actually, okay, we know that these were a distinct population from Neanderthals and from modern humans, but did they actually interbreed with modern humans? So this is just to give you the geographical distribution of Neanderthal and Denisovans. So as was in the previous figure, this is like the distribution of where we can find where Neanderthal lived, and this is like where actually Denisovan were thought to live. And this area is where we found the Denisova caves, where we have an area where actually Neanderthal and Denisovans were coexisting, co-living, basically. Okay, so what we know so far is actually that what I already said is that we have only one genome. So we can discover as much as only until a certain point with only one genome. So what has been done in in research is that actually what people have done is that they exploited the Denisovan DNA found in modern humans. So first I need to explain what is archaic introgression. So archaic introgression is basically what happened is that when modern humans moved outside Africa at a certain point, they encounter in Eurasia archaic humans. So we have modern humans outside Africa. When they encounter archaic humans and they interbred, and when they interbred, after you can imagine this is like their genome, that before modern humans genome was only green and archaic humans was only yellow. We have like we're trying to simplify very much, but when they mix, basically their genome looked something like that. Okay, a mixture of the two. So after many generations, what happened? This happens that actually archaic humans went extinct, so the admixture didn't happen anymore, and so this variation, the archaic variation found in modern day in modern humans genomes were diluted, and actually is found at low frequency in the genome. But there are two things actually, two exceptions. One are the so-called archaic deserts, so they are part of the genome in which we don't have archaic introgression, and there are other parts in which archaic introgression is archaic variants are at high frequency, and it is called this is what we call actually adaptive introgression. So this means that probably these variants were facilitated adaptation in some way, and we will talk about this later. So what I want to say now is that so scientists were able to understand that actually we have Denisovan ancestry present in humans, so Denisovan and modern day humans interbred. But the very interesting thing is that we don't find the highest amount of Denisovan ancestry in regions like in Siberia, you can say like in this region here, or in East Asia, but actually the highest percentage of Denisovan ancestry is found in Papua New Guinea or in like highlands of East Asia. So this area here, in which we don't have any evidence of the presence of Denisova in that so far in that area. And so exactly. So this is basically the map of the world with populations and the percentage of Denisovan ancestry that they bear. And you see, for example, in Africa, of course, it's 0, and then it's very, very low, basically almost 0 in Europe. And then you have a little bit more in populations such as East Asian and South Asians, and also in the Americas. But here, the amount of Denisovan ancestry is like around 5%. So it's very high. And this is very interesting. So these raised many questions. So why this population have such an high Denisovan ancestry is maybe because actually the area in which Denisovan lived is not only restricted to Asia, but was also in Oceania. So they were actually adapted to many different environments and so on. And actually, another question could be how many population of Denisovans existed at the time? Was this like different? There was structure in Denisovans or not? So, and yes, so this is basically the two. The geographic location of the two caves in which we have found Denisovans remains, and you see that are pretty far away from Oceania. Okay, so now let's try to understand. Okay, so did they interbred? We have this peculiar result of high frequency of Denisovan ancestry in population from Oceania. But actually, how many times this happened? How many times Denisovan interbred with modern humans? Was it the same Denisovan populations? Were different Denisovan populations? So what happened is that in the past, so I think from 2018, so in the past 6 years, there have been papers published that tried to that actually showed evidence that Denisovans interbred with modern humans multiple times. So now I'm going to try to explain these papers found. So the first one was in 2018, in which Browning and colleagues revealed for the first time that there were two pulses of archaic Denisovan mixture, in this case in East Asians. So they found two distinct Denisovan populations, found evidence of two distinct Denisovan populations that interbred with modern humans, with East Asians. Jacobs and colleagues in 2019, they found these two pulses of archaic Denisovan mixture, this time in Papuans. And again, in 2021, Choin and colleagues found a double a double pulse of Denisovan admixture in population from Oceania. And finally, in 2021, Larena and colleagues showed that actually there was a distinct population of Denisovan that introgressed with a group of Philippine Negritos. So if we count all these populations that actually scientists prove that interbred with modern humans, we have like a sum of 7 populations. But the thing is that we know that multiple populations introgress with modern humans, but each of these studies actually analyzed, used first of all different methods, different filters that I will explain later why this is possible. They analyzed, of course, different individuals because in some paper, like for example here, Jacobs published new data from Papua, in Choin published new data from multiple population in Oceania, and this new data from Philippines. So this we need to try to understand actually which are the things that are the same in this distinct paper. So what we try to do in the review is try to summarize these findings. And when we try to do that, we found that at least we can say that at least 4 populations from distinct Denisovan population introgress with modern humans. And now I'll try to explain why and how we find out this. So this is basically, imagine this AD is the Altai Denisovan, so it is the only genome that we have. And so we are comparing, of course, all this population to the only one genome, only high coverage genome that we have. And we can say that there are 4 populations that introgress with modern humans, and these 4 populations have distinct relatedness to the Altai Denisova. So some are more like this one called D0 is more closely related to the Altai Denisova, and like this one D2 is more distantly related. And we know this also because they tried to date the split to the Altai Denisova population. So for some is more relatedness, for other is less. So we'll see now actually where do we find these answers? Where do we find this population in modern day individuals? So we have, you see that this D0, that is the one more closely related, was found in East Asians. And D1, that is a bit less closely related, was found in Oceania. D3 is the one in the Philippines, and D2 was found both in Oceania and in Asia. So each of these papers found different evidence for this, and so they talk for the first time about one or the other Denisovan populations. So now we're gonna go one by one. So Brownings, they describe this true pulse of archaic Denisovan mixture in East Asians. So they found, so basically what they describe in their paper is these two, that they see these two distinct population, which is one is more closely related to the Altai Denisova, that is this D0, and the other one is more distantly related, that is D2. But how actually were they able to do this? So now I'm going to try to understand how they did that. So let's imagine that this is so these are modern human genomes, this line in gray. And this line here in blue is the sequence Denisovan, so our Altai Denisovan, the genome that we have, to which we can compare the modern human genomes. And these yellow bricks here are actually the archaic segments that have been found in modern human genomes. So what we can do is we can try to compare these segments to the sequence Denisovan, and so measure a match rate. Let's say this can be a measure of distance or similarities, depending how you look at it. So this is like the match rate to this segment here, and so on. So and then you can count these, of course, these segments in their entire genome. And so what we can do is that we can look at the distribution of these numbers, and this number actually can look something like that. They can look like there are two peaks. So what does it mean? So here you have on the x-axis the match to the Altai Denisovan, and on the y-axis the density. So how many times you find these numbers basically in these segments in the genome. And you see that there are two peaks because in this case, for example, we have a peak at 0.8 match rate. So here you have like an amount of segments that have been found at this match rate, but you have also an amount of segments of this match rate. And what does it mean? That there are segments that have a higher affinity with an higher match with the Altai Denisovan genomes, and others that have a less affinity, so a lower match to the Altai Denisovan genomes. So why is this? This has been interpreted as evidence of introgression events, basically. And this makes sense because what you expect, you don't expect that the actual donor of the Denisovan genomes, of the Denisovan fragments is the actual sequence that you have, so the Altai Denisovan sequence. But it can be a population of Denisovan that is more or less closely related to the sequence Denisovan, because what happens at a certain point, so there's been a split between these two Denisovan population, mutation happen, and so if this mutation happen, this population can be more or less closely related to the sequence Denisovan for many reasons. And so this is actually, for example, this is like a model to try to understand that this donor is the one that introduced the Denisovan segments, these Denisovan donors, into the ancestors of modern-day Oceania or East Asia and so on. So with this information, what we can do, for example, is infer the split time, infer this split time. However, this is not very easy because you need to be sure, if you want to do this, you need to be sure that these are actual Denisovan segments. So this requires to know that these are the real Denisovan segments. And this is not trivial because now I'm not going to be too technical in explaining this, but it's very complicated to find which are the real Denisovan segments. Usually you use, like in this case, a sequence, like a reference sequence, or usually you extract what you can find is there are generic archaic segments, and then you need to understand actually if these are Neanderthal or Denisovan, so you apply many filters. And so different papers, different scientists apply different filters, so this is not simple. But what has been done is that basically Browning, in this paper, they were the first one to apply this method, this very clever method of visualize the match rate. And this is called the contour plot. And basically what they did is that instead of measuring only the match rate to the sequence Denisovan, they also measured the match rate to the sequence Neanderthal. And you see here that, so in this case, you have the archaic segments, and you look at the match first to the Neanderthal, and then to the sequence Denisovan. So at the end, you obtain a plot like this, in which you have the match to the Altai Neanderthal on the x-axis, and to the Altai Denisovan on the y-axis. And these numbers inside the plot are basically, they represent the density. So higher the numbers, higher it means you have more segments at that specific match rate. And if you see here, basically what we can see is that this is like an example of an East Asian population and Chinese from Beijing. You see that for the match to Altai Denisovans, you have two peaks. So what does this mean? This means that these segments have a lower affinity to Altai Neanderthal, and the higher affinity, this is like around 0.6, let's say 0.5, and this is much higher, so to like 0.8, but so you have two different peaks. And so this is basically the distribution that we were seeing before. So this is how they were they were hypothesizing that in East Asians, you have there's been two pulses from two distinct Denisovan population because this population are distinct match to the sequence Altai Denisovan population. So this is to go back to what I was showing you before. So these are the two populations that you were seeing. So one that, like here, you have a more closely related to Altai Denisovan and one less closely related to Altai Denisovan. So the D0 is the one more closely related, and the D2 is the one more distantly related. So okay, so that was the first evidence of two introgression events from two distinct populations in East Asians. But lately, more then basically, Jacobs showed, first Jacobs, they showed also a double introgression event from two distinct populations also in Papuans and then in Oceania. And these two populations are these ones, so D1 and D2, where D1 contributed only to, it's found only in Oceania, in Papuan and Oceania populations, whereas D2 is the same population, and actually there are evidence that actually contributed also to East Asians. So, but it contributed also to Oceanian and Papua New Guinea. So these are like this is the figure from the paper when you can say, as I explained before, the two-peak distribution. You see here is like basically the same thing. So here you have the mismatch to the Denisova, and you see two peaks. So one D2 more closely related to Altai. Sorry, more distantly because this is the mismatch to the Altai Denisova, and D1 more closely related to Altai Denisova. And this is like the reconstruction of the tree, where you have here the sequence Altai Denisova. You have D0, that is the one only found in East Asia. So you see it contributes to East Asian populations. And you have D1 here found in Papua, more closely related, and D2 more distantly related. Okay, and finally, so was also shown by Larraín and colleagues that probably there has been a distinct population that also contributed to Philippine populations. And why did they think that? Because they show that this population from the Philippines, they have the highest amount of Denisovan ancestry in their genome, even more and much more actually than Papuan individuals. So Papuan are here, and you see this population have a much higher contribution. And so what they hypothesize is that this is not the same population that contributed to other populations, to other modern day populations, but is a distinct population. So what we can say is that we have, this is how we arrive to say that at least four distinct Denisovan population contributed to different modern humans populations. Okay, so just to do a recap of this part. So we can say this, so at least for different populations interbred with modern humans, but we have still many questions about this. So are there actually more population? Can we find out if there were more? Then a very important thing that I didn't mention yet, sorry, is why don't Papuans show two peaks in the contour plots? Because when I was showing before the contour plots of the East Asians, you remember there were two peaks here, but in the same paper, they didn't see that peak in these two peaks in Papuans, but instead they saw in a paper that was published later that there were at least two populations that contributed to Papuans. So this is like something that needs to be addressed, because we need to understand why there are these discordant results. And one thing it can be that actually we need to have better methodologies. We need new methodologies to try to actually identify the Denisovan segments. As I was telling you before, these these papers use different filters after when they find the archaic segments. They use different filters, so this can make the results different, and we need better methodologies to try to understand which actually are the Denisovan segments. Then we need also to analyze all the data collectively, because that could, of course, if you have a paper that published like 300 individuals more, we have more data, so it needs to be this needs to be done. And also, of course, we need new remains, new Denisovan sequence, because we base all against one Denisovan's genomes, and so it would be amazing to be able to find new remains, but also to sequence new genomes from Denisovan. Okay, so now I'm gonna go into the last part, and it is related to the biological impact of Denisovan introgression on modern humans. So if we go back to this figure, you remember that I was telling you about adaptive introgression. I want to say, so basically, this is very important. It's a very important to study, topic to study, because we have seen that Denisovans were found in different parts, different geographical areas. They're found in different in the genomes of different modern humans. So we want to understand if actually this Denisovan variation actually help modern humans to adapt to new environments, to pathogens, and so on. So what we try to do in the review is that we try to summarize this information. So to summarize the traits that have been adaptively introgressed from archaic humans. So here you have many traits divided by populations. And we can say that many of them are actually introgressed from Neanderthal, but there are some that have been introgressed from Denisovans, and this is what I will focus on. So first of all, I will, this was the first example of adaptation or archaic adaptations. So what they found is this paper is that they found this haplotype that is in the EPAS1 gene that was first of all, introgressed from Denisovan. So this is a Denisovan haplotype. Here you have the haplotype frequency, and you have population from all over the world, and these ones are Tibetans. And if you see, the Tibetans are the only one that have at a very high frequency the Denisovan haplotype, whereas other population from East Asia, they don't have, or from other population from all over the world. And so if you look at the haplotype distribution, here again, you have the Tibetans, the Denisovans, the Chinese, and the Neanderthal. The black are the derived alleles, and the white are the ancestral alleles. So you see that this haplotype is this one, is this part, these nucleotides, and this is like the same in Denisovan and Tibetan, but not in Chinese. So this was clearly, this is from Denisovan, and we find it only in Tibetans. And it's found in EPAS1 gene. But what is EPAS1? So EPAS1 genes encodes one subunit of a transcription factor that is involved in the induction of genes that are involved in the response to hypoxia. So when there's a low level of oxygen. So this I think it makes perfect sense that the population in this area of the world, so in Tibet, they needed to adapt to high altitude. And so this is a good example of a gene that actually derived from Denisova that helped them to adapt to this altitude. And so in another paper, so more recently, they tried to reconstruct the history and evolution of this Denisovan EPAS1 haplotype in Tibetans. And what they found very interesting is that this haplotype was introduced by the D0 population, so the one that is from the East Asian, to the ancestors of Tibetans around 48,000 years ago. But this selection didn't happen until 9,000 years ago. So this happened long after the introduction, the introgression event. And this is one example of what is called selection acting on standing archaic variation. So this happened much after the introgression event. So another example of adaptive introgression is related to metabolism. So in 2016, Rassimo and colleagues found that these TBX15 and WORS2 were introgressed in Inuit population from Greenland from Denisovan. And so this is just an example of one SNP in this haplotype that is fixed. Its frequency fixed in Greenland and is found in different frequencies in other populations in the world. And WORS2 WORS2 basically encodes for a synthetase, and TBX15 is very interesting because is an highly pleiotropic gene, is expressed in multiple tissue, but it also plays a role in the differentiation of adipocytes. And adipocytes, what they do is that they produce heat via lipid oxidation when they're stimulated by cold temperatures. So this basically makes TBX15 a very strong candidate gene for adaptation to life in the Arctic. Then it's very interesting that they also found WORS2 not only in Inuit but also in a following paper also in East Island Southeast Asian population. So you see here that it was found as selected and adapted as selected from Denisovan, of course. Okay. Then another example of adaptive introgression is related to immunity, and basically it was found that this gene TNFAIP3 is under selection in Melanesian population and is derived from Denisovan. And basically, this is a negative regulator of innate and adaptive immune response. So it's very important for tuning the risk of inflammatory disease. So it's related to, immunity response. And it was also found in Papuans and East Island Southeast Asia. Okay, now I want to talk briefly about this very interesting case that is, I think, a new case. So basically, I haven't talked about Denisovan introgression in the Americas so far. So what we know is that when modern humans arrived in the Americas, they spread from north to south very, very rapidly. So they had to adapt in some way to the different environments that they found when they moved. So an important question is actually did the archaic variation facilitated the adaptation in the Americas? This is a very understudied topic that we now know very little about, but we have one example. So what has been found in 2017 by Rassimo and colleagues is that there is this gene, MAC19, that is at high frequency in Mexicans. And this gene, this haplotype, derives from the Denisovan. You see here the dot is actually, it means that it derived from Denisovan. So here you have like the number of archaic alleles on the y-axis and the distribution, 95% quantile distribution of archaic and the rare allele frequency. So this is the method that allows you to find adaptively introgressed traits. So so this is like so high frequency in Mexicans. And what do we know about this gene? We know that this MAC19 is encodes for a mucin protein, and this serves as a defense against infection. So mucins are basically the most abundant macromolecules in mucus. So this can be very important. It's like a physical protection against external pathogens. So why this is also important is this because when so basically this work has been done by my colleagues and my PI, so Fernando and Dave and Emilia, that this study is now in review. So I hope you will see it very soon published because it's very interesting. And what they try to understand is actually who was the most likely donor of the MAC19 haplotype, because we know it comes from Denisovan, but when they looked more deeply into it, they understood that actually this was not from directly happened directly from an introgression from a Denisovan population. But what happened? So here you have a tree in which you have the Altai Denisovan, the three Neanderthals high-coverage genomes, and modern humans populations. And what they found out is that probably these haplotypes was introgressed first from a Denisovan population that was not, of course, the Altai Denisovan that we have sequenced, but another population of Denisovan into the ancestors of Chargiskaya and India Neanderthal that are so-called late Neanderthals, and then introduced the ancestors of East Asians and Americans. So when the encounter of after the out of Africa. So what happened here is that this is another example of selection that happened only after a while. So this is another example of selection acting on standing archaic variation. So this we don't find this in East Asians. We find it only in Americans at an high frequency. So this is a new and interesting example of selection acting on standing archaic variation, basically. So just to summarize, these are I've tried to show you which are the example and the evidence that we have for adaptive introgression. So we have different traits in different population. We have population like in Tibet, when there's been adaptation again for hypoxia, favor of hypoxia tolerance. In Inuit, we have genes involved in lipid metabolism. In Papua New Guinea, we have immunity-related genes also, as well as also in South and East Asians, and this new immunity trait in the Americas. So what are actually, if to conclude, let's see actually what are the current challenges and open questions. But first, let's see. So what we saw in the seminar is that we can say that Denisovan were structured populations. So we have at least what we can say so far is that we have at least 4 distinct Denisovan populations that are distinctly related to the one genome we have, to the Altai Denisovan, that contributed to different modern humans populations, and at different time and different scales. And we have also examples of clear examples of adaptation favored by archaic introgression. So we know that Denisovans are still, we have this example, but actually Denisovans are still a very mysterious group because we have done this all with only one genome. So it might be that if we have more genomes, the story might change. We can have more evidence of in favor of some hypotheses, and we can have other theories. So it's a field that is still very open. And so what are actually the current challenges? So we need to study Denisovan in different populations because so far we studied it mainly in Papuans, in Oceanians, and in East Asians, because these are the population in which the Denisovan ancestry is more present. But we need to study it more in the Americas because there is a little bit of Denisovan ancestry there, and also in South Asian. So this might bring new, as you've seen, for example, for the MAC19 example, it might bring new evidence for or new knowledge about it. Then we need to develop new technologies to be able to detect the Denisovan fragment, to be sure that what we're analyzing is actually the Denisovan fragment. And so this will help to find new results. And then, of course, we need to find new Denisovan remains. There are some, but it's always very difficult to extract DNA from archaic remains or from ancient DNA, or sorry, from ancient remains. So even new techniques that allow to sequence ancient DNA will be helpful. And so, of course, this will definitely be useful for the field. And so probably there are even more open questions and challenges that now I'm not reporting, but I'm sure there are still many. So now, first of all, I want to thank again Phoebe Rachel for inviting me. I want to thank my PI Emilia for everything, my colleagues in Trinity, and my colleagues in Brown, everything, and you, of course, for being here and listening to me.
Thank you so much, Linda. That was really fantastic, really thorough.
Thank you.
Do you want to unshare your screen?
Hmm.
And then if
Yes.
anybody has any questions, as I said before, you're very welcome to either put them in the chat and I will read them out, or you can put your hand up and once I say your name, go ahead and unmute. Just give it a second and it takes some time to type stuff. We've got a question from Andrew
Yes.
in the chat. You said that there was only one Denisovan genome, but the bones in Bashir cast were also Denisovan. On what basis was that decided?
Yeah, so basically there's no whole genome from those remains from that mandible. There's the first paper I think that they published was about a proteomic ancient proteomic analysis in which they found out that that was a Denisovan, and later paper from sediment they were able to find mitochondrial DNA that belonged from that cave that belonged to Denisovans, so proving that in that cave they were living Denisovans.
Yeah, that makes sense.
Yeah, hopefully we will find also more remains and to prove that with the whole genome from that cave. That would be great.
Yeah, definitely. We've got two more questions. I'll ask first another one from Andrew. This is a good question. Why do you think there are so few remains of Denisovans?
That's a good question. I don't know. I think yeah, that's tough. More from an archaeology point of view, I don't really know. Maybe also the population were little, so it was more complicated to, they were not preserved. It's more difficult to find them. That's a good question, yes. Because, yeah, we hypothesized that actually they lived in not only in Asia, but also in Oceania, but there are no evidence so far. I think there are evidence also, there start to be a morphological evidence in Laos, for example, or other, but it's still East Asia, so we don't have anything else. But yeah, that's a good question.
Yeah, thanks, Andrew. We've got a question from Alejandra saying, great talk. Thank you so much. What does the presence of Denisovan genes on modern humans tell us about the adaptation, biogeography, and lives of the Denisovans themselves?
Okay, yes. So I think what we can say is that they were probably adapted to many different environments. So they definitely, except for like, for example, the EPAS one or the MAC19, that it seems to be an adaptation that happened later. So not in the Denisovan genomes, but in modern humans genomes. There were some that actually probably were already present in Denisovan. So they were definitely adapted to, life in Siberia, but life also in East Asia, in Southeast Asia. So we know that they were living in these different environments and they were probably adapted also to different pathogens. So that what we can hypothesize that were a population that were found in different areas. So that help, of course, that's the things that we know from a modern human genome is more than what we know from the single genome that we have. So that's from where we can hypothesize. And then
Thanks. Another question. This one is from Johan. I believe there are introgression deserts in brain-related genes. Is any work being done on the implications of such deserts?
that I'm actually not sure. I'm sorry, I cannot. I don't know about this. It might be because there are. Yes, there are introgression deserts in many parts of and I think people are investigating this, but I'm not really. I don't want to say anything that I'm not sure about. So I'm sorry.
Yeah, no, that's fair enough. Thanks.
It's interesting, but I don't know.
hmm We've got a comment
Yeah.
from Chris who
Yeah, I
I think
agree.
this is following up on another one,
Mm-mm.
on one of the earlier questions. I think it's quite likely that more of the Chinese fossils are Denisovans. Morphological studies already suggest that, e.g. Harbin and Yunxian studies, and more evidence is coming.
Yeah, definitely. Yeah, exactly. From morphology, we have some suggestions that these are like they were more the way the Denisovan were living in different areas. And yes, from genetics, this is what I can tell. And then we'll see, hopefully, to have both both evidence to compare. And yes, let's see.
Yeah, thanks for that. We've probably got time for one more question, if anybody has another. I'll give it a minute or so. Well, I can't see anything at the moment, and that's taken us quite nicely up to 2 p.m. So thank you so much for such a
No, thank
wonderful
you.
talk. It's been a really wonderful way to end the seminar series for this semester, and I found it really, really interesting, as I'm sure everybody else did too. Yeah,
Thank
lots
you.
of thank yous in the chat.
Thank you to all for coming and for you to inviting me.
Yes, indeed. Thank you, everybody, also for coming and also enjoying this talk along with me. Goodbye.
Bye.